Welcome to a deep dive into the world of veterinary neurology and rehabilitation, as we explore a cutting-edge study on surface electromyography or sEMG in dogs. This research, published in Methods X in 2024, investigates the use of sEMG to assess muscle function in dogs, particularly those suffering from intervertebral disc disease or IVDD. This condition is a common cause of spinal cord injury in certain dog breeds and can lead to pain, motor impairments, and neurological deficits. The study highlights the pressing need for objective assessment tools in veterinary rehabilitation to track patient progress. Traditional methods often fall short in providing detailed insights into neuromuscular health. This is where surface electromyography comes into the picture, offering a non-invasive way to measure and analyze the electrical activity of muscles. sEMG allows for evaluation of the shape, duration, phases and amplitude of electrical impulses, which helps in identifying pathological conditions, like chronic denervation. While sEMG is well established in human medicine, its application in veterinary practice is still under development. This research seeks to explore the possibilities of using sEMG to assess neuromuscular function in dogs with IVDD, focusing on the muscles of the thoracolumbar spinal region, which is highly susceptible to this disease. The study asks three key research questions. Firstly, can a device be developed for sEMG data acquisition in chondrodystrophic breeds, which are predisposed to IVDD? Secondly, what experimental setup and protocol can be used to evaluate neuromuscular function? And thirdly, can sEMG be a valuable tool for assessing neuromuscular function in dachshunds with IVDD in the thoracolumbar spinal region? Now, let's delve into the specifics of the study design. The study included 4 miniature dachshunds, aged 3 to 7 years, that attended the Anikura Restilo Veterinary Rehabilitation Center. 2 dogs were healthy and had no clinical signs of neurological or orthopedic diseases. The other 2 had been diagnosed with IVDD through CT scans, and had undergone surgery and rehabilitation sessions. This arrangement allowed for a comparison of sEMG data between a control group and a group affected by IVDD. The researchers modified a SDR device to acquire the sEMG data. This device was equipped with 3 electrodes, 2 differential leads and 1 reference lead, and it also had a 3-axis accelerometer for motion sensing. The sEMG sensor was designed to measure voltages between 0 and 3.3 mV with a very high amplitude resolution. The device was also protected by a 3D printed enclosure, as shown in Figure 2, to ensure that it could be easily and securely attached to the dog. Before applying the electrodes, the dog's hair was clipped in 3 small, square areas, each measuring 1.5 by 1.5 centimeters. 2 electrodes were placed in the paraspinal region, one between the thoracic vertebra 12 or T12 and the lumbar vertebra 2 or L2. These 2 were 1 centimeter apart. The third, or reference electrode, was placed on the medial aspect of the elbow. These areas were then cleaned with alcohol to reduce impedance, which could affect the signal quality. The sensor was fixed to the dog's torso with an adhesive bandage as seen in Figure 3, ensuring all 3 leads were connected to the correct anatomical sites. To evaluate muscle activity, the researchers used a protocol involving several exercises. These included walking on a treadmill at 0.7 miles per hour, which was further divided into accelerating, decelerating, and stable walking sub-movements, balancing on a wobble board, performing an isometric exercise with front paws on a donut ball, and standing still. Each activity was performed for at least 1 minute, and the movements were recorded by video to be matched with the sEMG signals. The sEMG signals were acquired via Bluetooth and transmitted in real time to a computer. An audio cue was used to synchronize the video recording with the signal data, helping with post-processing. Let's move on to the signal processing. The raw sEMG data was filtered using a 6th-order bandpass digital filter with a frequency range of 24 to 500 Hz to focus on the relevant frequencies for EMG, as noted by Conrad. In contrast to the amplitude, the mean frequency, or FMean, as shown in Figure 5b, revealed that the IVDD group consistently had higher values across all submovements. The treadmill exercises showed consistent values within each group, and the donut ball exercise, or submovement D, had the highest mean frequency in both groups. The largest difference in mean frequency between the 2 groups was observed during the wobble board exercise, while the smallest difference was during the standing still segments. Table 2 provides a detailed breakdown of the numerical values of these measurements, showing the mean, standard deviation, RMSA, power integral, Fmax, Fmean, and Fmedian for both groups across all activities. The data revealed a trend that supported the researchers' hypothesis. In dynamic exercises like the treadmill walking or wobble board balancing, there was a decrease in sEMG amplitude and an increase in sEMG frequency in the dogs with IVDD. Now, let's turn to the discussion of these findings. The study supports the idea that variations in muscle function due to nerve compression from IVDD can lead to lower sEMG amplitudes. This is due to the abnormal muscle activation patterns caused by muscle denervation. When the muscles don't receive the necessary neural stimulation for normal contraction, neurogenic atrophy occurs. As a result of denervation, there is a loss of motor units or MUs, which are the muscular areas innervated by a single motor neuron. This loss results in a reduction of the recorded sEMG amplitude, because fewer muscle units are being stimulated, which is indicative of a neuropathological condition. The changes in frequency can also be attributed to nerve damage. Nerve compression can accelerate muscle fatigue, which in turn affects the sEMG signal characteristics. Initially, during fatigue, there may be an increase in sEMG frequency. However, as fatigue progresses, the sEMG amplitude tends to increase while frequency may decrease. However, the exercises in this study were short and not physically demanding enough to induce late-stage fatigue. Additionally, denervation often results in muscle fibrillation, leading to small and erratic electrical impulses. These abnormal firing patterns of motor units can lead to higher sEMG frequencies. These findings are consistent with previous research by Okuno and Arito, Lee et al., and McKay et al., who reported similar results related to the impact of spinal cord injury and muscle denervation on sEMG signals. Overall, the results of this study support the potential use of sEMG as a diagnostic tool in veterinary medicine. The study also highlights the importance of using the root mean square amplitude or RMSA and the mean frequency or Fmean, as they are the most representative parameters for assessing muscle function based on Lowery and O'Malley and Petrovsky. The researchers also mentioned the use of a 3-axis accelerometer and suggested this data could be used in the future to assess the biomechanics of the animal in greater detail. This could provide further insights into the effects of IVDD on mobility and muscle function. Now let's address some of the limitations of this pilot study. The researchers noted difficulties in skin preparation and electrode placement, as well as some reluctance from owners to cut their animals' hair. They proposed that exploring dry electrodes adapted to veterinary medicine would be beneficial, as this would make the process of signal acquisition easier and not cause any aesthetic issues related to animal hair. The study also had a limited sample size with only 2 dogs in each group. Because of this, they emphasized the importance of conducting further research on a more extensive scale to obtain statistically significant results. Even though the sample size was small, the results suggest a possible trend warranting further study. The researchers state that a new electrode design that wouldn't need for clipping would also be beneficial to recruitment of more animals into research programs. In summary, this work demonstrates the potential of using sEMG as an objective tool for assessing neuromuscular function in dogs, particularly those affected by IVDD. The research also addresses the need for standardized procedures in veterinary sEMG, such as the electrode placement, skin preparation, and the use of specific exercises for data collection. The findings from this study could lead to improved methods for monitoring patient progress in veterinary rehabilitation medicine. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.